Hello. For my science experimental project, I will demonstrate how to extract DNA from a strawberry. But how does it work? Well, DNA is located in a cell's nucleus. Its purpose is to store information and hold instructions to make other parts of a cell, such as the creation of RNA or transcription. It's then translated into proteins. The action of translation lets these important instructions from DNA help the process of giving life to cells and lets them function properly. Fun. For this particular experiment, you will need some rubbing alcohol, which is in the freezer. We have strawberries, but you can also do this with bananas. Some dish soap, a lovely Dawn dish soap. We also need some salt and water, some sort of cup. We're using 2 disposable red Solo cups because I don't want to wash them. We need some sort of filter. Using a sifter to be environmentally friendly. And then you need a stir stick and I guess a knife to cut off the little bits of strawberry leaves and a trusty plastic bag. Right, now we're gonna get our strawberries. We're gonna get about, I don't know, maybe 4. Maybe 5. That one was a little small. Lovely. Now we're gonna take off any little extra green bits, especially this one. Look at that stem. Alright, we're gonna chop it up. Alright, now we're gonna put all of our strawberries in our plastic bag. So, fun fact, the DNA lives inside the nucleus. So while smushing the strawberries, we're breaking the cell wall's cell membrane and nuclear membrane, which releases the strawberries' DNA. Alright, yeah, so switching over, we're gonna bring it to our cup. So we're gonna add 2 tablespoons of dish soap. Dish soap. Cup. Lovely. Smells great. Smells great. I'm gonna add our 2nd tablespoon. That's right, you heard me right. 2 tablespoons of dish soap. Wasn't that nice? Alright, now we're gonna add 1 teaspoon of salt. I'm gonna put this over there. Teaspoon of salt. Look at that. Nice big old teaspoon. Gross. Don't eat that. Here, you get to stir with your stick. I love sticks. You can't see that. Cool. So here, the dish soap is being used to break down the cell membrane. Alright, we're back. Because I'm using a sifter, I decided it would be way easier if I used a table. So we're gonna put all of our strawberry concoction into this here filter, as you can see here. And I'll let you watch while I do my thing. This looks so gross. This is good content. Alright. Yeah, I'll let you guys do. Doesn't that look fun? Cool. I'm just gonna kind of mix around with the spoon, get all the clean spoon. Important. Just gonna make sure all the liquid I can get in this here bowl. Alright, check it out. Alright, time to add our 100 milliliters of rubbing alcohol into our bowl. interior, as you can see, it's making like kind of a cloudy sensation. You see, like kind of like right there, like that. Super, super cool. And because it's so cold, the rubbing alcohol, it makes the process of DNA separation faster. The rubbing alcohol used in this process is because the DNA is not soluble in it as it is to water. The DNA strands tend to float a bit, which lets the DNA come up into the rubbing alcohol layer. It's important to not mix it after a couple seconds. You want to make sure to wait a hot second, watch as the white cloudy substance forms onto the top layer. After a few seconds, you can start mixing it. Like me right now. I'm gonna get a stick. And I'm gonna start mixing. I love mixing. And now we can pluck out the strawberry. Whoa. Huh. That's gross. I mean, it's pretty cool, but no, I'm not a huge slime person, but you know, for an experiment, it's fun. It's cool. It's quirky. What a journey, am I right?